Muscle tissue. This is the one of the four basic tissues in the human body. Contractility is the fundamental property of all animal cells, but it attains its expression in muscle cells. Greatest to shorten effectively, the muscle cells are long and narrow and thus called muscle fibers. Its ends are usually attached to a bone, and from it delicate sheets of connective tissue provide all these three fibers coverings: EPI, peri, endo, transmit blood vessels, nerves, extensive network of capillaries. The outer surface of the power. Substantial amount of glycogen, distributed throughout the cytoplasm, while isotropic band is monorefringent and does not alter the plane of the polarized light. 34A striated, skeletal or voluntary muscle. Shows striation under microscope. Ost constant aisle coverings muscle. Mention they striated muscle TSGEWRIN. The entire muscle is covered by tough sheath of relatively dense connective tissue AD called as coenzyme. From zyme fibrous partitions extend into the muscle to surround fascicle bundles of mucil fibers. These are called as perimysium. Network extending between cache and every muscle fiber and constitute endomysium. Lymphatics into the muscle. Endomysium shows fibroblast. Collagen fibers and the cut muscle fibers show peripheral nucleus. Striated muscle LS1, P. Muscle fibers are large, cylindrical. They show relatively large number of nuclei multinucleated, which are ovoid and elongated with long axis parallel to the muscle fiber. They lie just beneath the sarcolemma. The nuclei of fibroblast in the endomysium lie close carcolemma. Cytoplasm is acidophilic and cross striations are not always clearly seen under low. In addition to the myofibrils, the cytoplasm contains numerous mitochondria, and smooth endoplasmic reticulum is extensive and is called as sarcoplasmic reticulum. Fill striated mucil loss, HP. The cytoplasm is filled with numerous longitudinal fibrils called myofibrils, uniformly striations or alternate dark and light hands, also called as A anisotrophic and I isotropic bands respectively. Anisotrophic means I doubly refracts light, i.e. alters the polarized light in planes. These striations are better seen in polarized light. Band shows Z lines in the center and a band shows lighter H zone in the dark band. The center of the light H zone shows a dark M band line. A rusty earth legson. Umbilical. The striated muscle. They are of two types, actin and myosin. They are made up of actin and myosin proteins respectively. Is free in the I band and also extends in the outer parts of a band where the myosin filaments are confined to the A band equal to the width of the A band. H band is named after Henson, the part of the A band into which the actin filaments do not extend. In the relaxed condition, uncontracted, the overlapping of the actin and myosin is minimal. During muscle contraction, two Z lines come closer due to sliding in of actin filaments into the intervals between the myosin filaments. The width of a band remains unchanged, but the width of the I band decreases. The H band disappears in fully contracted sarcomere. Myosin filaments establish bonds with adjoining actin filaments and head of myosin bends due to bond. This bonding drags the actin with head of myosin, then unbends and establishes another bond with the next part of the actin filament. These bonds are in the intervals made in unmade rapid succession, dragging the actin filament between the myosin, leading to muscle contraction. Each actin filament has a head end that extends into the A band and tail end that is anchored to the Z line. Sarcomere is the part of a myofibril between two consecutive Z lines. Sarcomere is approximately 2 or 3 U in length and is the ultimate contractile unit. The line is at the junction of two sarcomeres. A sarcomere relaxed. Each myofibril is made up of myofilaments. Actin is thin filament with 8 nanometers diameter. Actin filaments are attached to Z band, German Zuichenschieb, between the C through actin and protein present in the Z band. The second end of the actin filament interdigitate with myosin filament. I band is made up of only actin filaments alone. Myosin is the thick filament with 12 nanometers diameter, i.e. the length the myosin filament is M band, German Mittelschieb equals middle, seen due to fine interconnections between adjacent myosin filaments. B. Sarcomere _ contracted. Two twisted subfilaments of actin are made up of globular molecules of G-actin. Large number of myosin molecules form myosin filament. Each molecule has two subunits and cache unit has a head and tail. All the heads project outside the filaments and tails are attached to the M line. Out into bubble-like expansions. Lines are absent. It is present in the heart. Out it is striated but not under voluntary control. The structure of the sarcomere is same as that in the skeletal muscle. Strictly parallel, but they branch and anastomose. Each muscle cell is about 80 long and 15 in diameter with single centroskeletal muscles. The slit-like spaces, the junctions between adjacent muscle cells, seen as dark staining transverse lines, they may appear broken into number of steps. These discs lie opposite the I band of the striations. The cytoplasm near the cell membrane junctions shows dense appearance with actin filaments embedded in it. 38B smooth non-striated or involuntary muscle. PI smooth muscle fibrae single cell with single central nucleus. Livers of smoth muscles are commonly subdivided into bundles of Brie. The ends of fiber in bundles interdigitate with those of other bundles. Muscle fibers elongated and tapered at the ends, having length 30 pt 500 grams. Average length is 200 y with SP diameter, while longest are seen in pregnant utens. The nucleus lies in the widest part of the fiber, i.e. in the middle. The cytoplasm is eosinophilic and uniform. Relaxed smooth muscle is spindle-shaped with smooth outline, while contra muscle assumes a more ellipsoid shape in the cell membrane and cytoplasm. B. They lack striations because filaments overlap each other in no definite pattern. N. See cardiac muscle. Muscle fibers are not network or syncytium. Formy nucleus. The myofibrils are relatively few so that the striations are not as distinct as between anastomosing fibers. Contain endomysium that carns capillaries and lymphatics close to the muscle fibers. These spaces show the nucleus fibroblasts. M. Cytoplasm of the cardiac fiber is eosinophilic. Intercalated discs HP running across the fibers are called intercalated discs. Sao, layer of vested with connective tissue supplying if with capillaries. And mineral co-tiadeth dispam 9 spurs though brutine. There are numerous desmosomes, gap junctions and tight junctions. Gap junctions allow electrical continuity, thus cardiac muscle syncytium but not anatomical. 1s. Physiological.